I'd like to thank the organizers, Pavel and Sasha and others, for inviting me to this very beautiful venue. For those of you who have known Pavel for a long time, actually probably know that he's always been insistent on clean formulations of problems. And I thought I might show you a slide from almost 20 years ago. It's always bad to put up these slides because it kind of dates you. A paper that Pevsner and Waterman wrote, which said open combinatorial problems in computational molecular biology. And in fact, if you go through the paper, you'll see a whole bunch of problems on genome rearrangements, and it's fun to go and see how many of these have been resolved in the years since, and by many of the researchers who are here. So of course, the field of genome rearrangements has grown well beyond those initial formulations, and of course now there are books out on the subject, and you heard this very nice talk from Max Alexeyev, sort of coming out to the state of the art in this field. Now we should also think about formulations from a different perspective. It used to be, again, many years back, that we were data poor. And in fact, here's another paper that I pulled out from many years back, also written by people who are in this room, Dick Carr, Pavel Pevsner. And it argued that actually people who do computational biology and other fields should really pay attention to the applications and to look at the data so that they don't formulate problems that actually don't have any meaning in the real world. And of course, if you think about it, we have really come full circle, because we are in such a data-rich environment now that people often forget that they need to formulate the problem cleanly, and I'll take some examples as we go along. So I think this conference is indeed quite timely. So let's go to the data and actually talk a little bit about what causes these kinds of rearrangements, the ones that Max talked about, and of course others. So here's one plausible mechanism by which these can happen, and they make it sort of repeat mediated. So of course, in our genomes, we have repetitive sequence, and when the two genomes line up just prior to sort of cell division, the repeats might not line up in order. And so a rearrangement operation might take this part of the genome and join it with this. And when that happens, you might end up with a recombined genome that looks quite different from the two parental chromosomes, right? In this case, this particular crossover caused a duplication of this region. So you have an example of a rearrangement. And of course, it could happen the other way also. And in that case, you would end up with a region, with a genome that tends to have this region deleted out. And sort of, if your repeats were inverted, you could have inversions, much like what Max talked about. Right? So of course, in the data poor environment that we were in those days, we sort of imagine that these events were probably very rare, because a deletion, for example, a 10,000 base pair deletion or 100,000 base pair deletion could easily delete a lot of genes, making the organism not viable. And so these events were rare, and once in a while they would survive and maybe even get fixed in a population, and then therefore act as a phylogenetic marker, and hence the original use of rearrangements in estimating phylogenies. But when you start looking at the data, of course, the situation changes completely. It turns out that these rearrangements are not only not rare, they're actually very common. They're so common that when you sample a genome, and this is a paper from a few years back, you see a huge amount of rearrangements relative to another human reference. So even within a species, when we look at different genomes, you will see a huge variety of insertions, deletions, inversions. Almost everything that you could expect to see, you will find just within a species. And therefore, the problem also changes in character. Rather than just using these rearrangements as markers for genomes, we could use them to study the variation in a population, or maybe in the case of disease, to see the genealogy, the etiology of the disease as a function of rearrangements. And so, of course, there's been a line of work along this. But one of the classic cases where you see tremendous amounts of rearrangements are in cancer. And here, people have now begun to question the fact whether these simple models that we considered beforehand, like inversions, duplications, deletions might even be sufficient to explain cancer. Of course, Ron is in the room, and he's done some really beautiful work explaining how one could take a sequence of these rearrangements and see the evolution of cancer genomes in that context. But when you look at data like this, and the way to understand this is the genome, a chromosome laid out end to end, and you have copy numbers of the chromosomes. So here, the number of copies is increased relative here, where here the copy number is decreased, and here it's all stable. So you can see that there's this tremendous amount of rearrangement that has occurred in this genome. Here's another example from a different cell line. And here you see, if this is a logarithmic scale, so you see a tremendous expansion of this region. When you look at rearrangements like this, sometimes you do question if the simple rearrangements that we talked about before can actually explain this kind of complexity. And that problem is not completely solved. But again, a few years back, some researchers, and this is from the Stratton group in Cambridge, which is sort of the preeminent group in cancer genomics. They have just many, many path-breaking papers. And they claim that this is indeed the case, that what they see out here in their data is that it's not simple rearrangements, but more a kind of breakage fusion bridge cycle, which was originally proposed back in 1941 by Barbara McClintock. And they claim that this is the first time that this has been observed in cancer, and these kinds of exotic rearrangements are actually what's causing the rearrangement. And if you look at their subsequent papers, they have come up with even more exotic models than this one. This is what we'll talk about today, like chromotripsis and many others. And then there's this entire there's this entire sort of body of work which shows evidence for all these rearrangements. Now it's funny because we started from this data poor environment where formulation was key, and now we are in this data rich environment. And if you think about it, these very distinguished scientists definitely failed the Pevsner test, which is that they explained a solution, but they forgot to define the problem. And this might seem a little bit sort of unusual to people in this room, most of whom are very well trained in mathematics and so on. But we do have a graduate program in bioinformatics, and Pavel teaches a class where he encourages and I would say asks people to formulate problems. And I have had so many students come to me and say, and these are very smart students, but they are more from the biology side. They say, we have this nice problem, and we have a beautiful solution to it, but we cannot formulate it, and they cannot pass the class. And so here's another example of this where they say that this is due to breakage fusion bridge, but they didn't quite explain the model. So I think that's what we set out to do in this talk. And in order to explain this in a more formal setting, we do have to go back all the way to Barbara McClintock's very seminal work. Of course, she was a very famous scientist. She worked on, if you've heard of jumping genes and transposons, for which she later got the Nobel Prize. Her work is very well cited. But it remains now, as it was then, quite misunderstood. And for those of you who know the history, she spent a lot of time being a woman in academic positions without actually having a tenure track position. And sometime in the 40s, she was at the University of Missouri. Again, she was a professor, but never invited to any of the faculty meetings. And she spent sort of her time in complete isolation, where people were hiring, and many times they were hiring other women faculty, but she was not invited to participate in these discussions. And this is when she came out with this really beautiful idea of an operation that she observed simply by looking at maize chromosomes under a microscope. So what happens is that periodically there might be a break at the telomeric end of the chromosome. Okay? And when this happens, the telomere, of course, is a great stabilizing agent. When this break happens, it kind of makes the chromosome unstable. And so when the sister chromatid is formed, just in order to stabilize the chromosome, they form a bridge out here. And so we have this breakage, and then there's the fusion bridge. Okay? But these two chromatids must separate during cell division. And so when that happens, there's a stress on this chromosome. So here I've only drawn till the centromeric part. So here are the two centromeres, and they are being pulled apart. And so there's stress on the chromosome, and they will break at some point. And so when this happens, of course, the chromosome kind of breaks apart. And what you get is the original chromosome plus a little part of the chromosome that both got inverted and duplicated as well as inverted, right? So that's what a single rearrangement would do to you. But now we again have an example of an unstable chromosome because this part is broken up. And so the same operation can repeat. You have another sequence of breakage, fusion, bridge, and it shows up as an inverted prefix duplication, and then another, and then another. And what you end up with is this very complex rearranged form of the genome. All right. Now, of course, one might say, well, what does this have to do with biology? Today, it was some odd thing that she observed under a microscope for the maize genome. Turns out that telomere instability is, of course, a major driver for cancer because it makes the genomes very unstable and causes them to rearrange in this peculiar ways. And sometimes they can sort of amplify oncogenes and therefore lead to cancer. And here's sort of an example from just last month where people, they did a hybridization experiment where they took two centromeric regions, and when you can see them, the two centromeres come very close together, and that allows you to see that there's a fusion going on. And here's another example. Here are the two centromeres, and here's the fusion going on. So you can actually see these fusions under a microscope, similar to what Barbara McClintock did, but slightly more, just slightly better resolution. Okay, and now what was claimed, of course, by Stratton and colleagues is that not by looking at cytogenetic evidence, which only gives you evidence of fusion, but not really the kinds of rearrangements that you see, but more molecular evidence, array hybridization and sequence data, that they could look at that evidence and say that breakage fusion bridge was happening. What does that mean for us? So we are not at the stage where we can assemble these genomes. You've heard some beautiful talks on assembly, and of course, the major challenge with assembling genomes is the presence of repeat elements. When you have genomes like this, where there's a lot of repetition, you cannot take fragments and assemble them. At least we don't have the long reach to assemble them fully. But you can, sample them. You can sort of sequence them or hybridize them, and you can measure counts of separate segments. So here, I'm sorry, this is not very visible, but you can see that A appears 4 times, B appears 4 times, C appears 6 times, and you get a sequence of counts. That's the kind of data that you expect to see. And one very simple formulation of this problem could be simply this, that if you're given a segmentation of the genome with copy counts, and you can do this, you can get this kind of data from existing resources like array hybridization and sequencing. The question is if this set of counts consistent with a BFB sequence? That is, can you describe a sequence which only contains these letters, maybe repeated many times, such that that sequence would have arisen from a sequence of breakage, fusion, bridge operations, and that the counts satisfy what's out here. And in this case, if we already showed that this was, this was BFB, but if you take this count and you reduce it by 1, it becomes not BFB. The problem turns out to be harder. Yes, Mike? Yes, because the original order is, from the centromere to the telomere, it's always in that same direction. And once you do the segmentation, then you know the order.
Does BFB comes with the loss of fragments?
I'm sorry, loss of fragments? Yeah, so I didn't say this, and I think I'm going to skip that part. There are two ways, whenever the two chromosomes split apart, one side is longer, the other is shorter. And so if you have a sequence of events, sometimes they are shorter and sometimes they are longer. You can sort of prove that you can always ignore the shorter ones. And the case where you have a deletion and you come up with an equivalent representation that doesn't lose any of the originality. So you can always imagine that as the genome grows. But what you have, if you have a mix in a population and a mix of shorter and longer, then that could be a bit tricky. All right? Yes?
You're assuming that BFBs are the only things that are going on.
I am not assuming that. There is no such assumption. The question, in this particular case, in this formulation, yes. But when we actually go back to the data, as was said, we will use BFB against a competing array of transposition. And that's where the difference between the clean formulation and the actual use of this comes in. But thank you.
You also get the number of consecutive segments. So can you get how many times A and B come together?
Yes. So what Pavel asked is a very good question. He says, is this the only kind of data that you have? When you have paired end sequencing, sometimes you can get a sense of junctions. And so when you have breakage fusion bridge, you have A minus A junctions. And yes, the answer is yes, you can. And that changes the formulation a little bit, and it helps. And I'll show you some data to show that as well. Yes. All right, very nice. So we have now a clean formulation. It turns out that this formulation is not as easy to solve. We came out with a partial solution sometime back, and now today I'll present a full solution to this problem. It did turn out to be much harder than when we first started. So here's the same. I've drawn the chromosome here. I'm sorry if this is not visible. I've just written A, B, and C, but I made it into a graph where C points to A and then B points to A. And let's say that the first breakage fusion bridge happens between B and C, and so A and B get duplicated. And I always write them in the same order, but the arrow shows that this entire part is being replicated and mirrored. All right, and now we do a second operation, and this time it's between, it's between A and B, so this part is getting duplicated, and you can just see, do it by, imagine a reflecting surface here and then just reflecting it out here. And then you go on for a little while. And then finally you can do an operation that duplicates everything together. And that without loss of generality, this last step you can always do so that you get some symmetry. Something that starts at C always returns back to C. And we will use this kind of formulation. All right? So now when you think about it from this perspective, what you're given is the input counts, and the goal is to create this path that you can, that would explain a BFB sequence explaining these counts. And when you see the data like this, you can immediately see that there's an exponential time algorithm, because at every position in the node, you have at most two possibilities. You can either go right or left, and that depends on what level you are at, or you can go up. And so one of these two possibilities, and you have 2 to the n ways of trying out paths that will fit this. But remember, the size of the data is only k log n, so it's sort of doubly exponential. So in order to speed it up, we came up with a slightly different, it's also a heuristic, but it does quite well in practice. So what you can do is, you think about the time that you start from a particular letter, and the first time you come back to it, and you glue them together. Right? So in this case, the 2 C's, you glue them together. At the next level, you have many B's, but you take the adjacent ones. The first time you start, you return, you glue these, then these, then these, and these. And then you do it for A as well. And when you do it this way, you can sort of make a tree-based structure where this particular thing is the node out here. And then there are 5 nodes corresponding to B, half of the total number of B symbols. And then 6 corresponding to A. And the paths, the sort of subtrees represent the hierarchy that you see out here, right? So this is what we call a BFB tree. It's a symmetric tree, and there are certain symmetries which actually allow you to completely characterize what BFB trees are. And given such a tree, you can also derive a sequence by just sort of going in an in-order fashion and just reading out the letters for every level. So the letter here is C, the letter here is B. If you do a kind of in-order traversal, you will come back to the BFB sequence. Right? And so essentially, a BFB tree is a tree with certain symmetries. The first is that if you take any node, you can see that there's a symmetry. That means that if you rotate around this node, you get the same tree again. And likewise, there's what we call a paired end symmetry. That is, if you take 2 nodes at the same level and consecutive, then the subtree that is induced by traversing this path, that is also symmetric. Right? And then finally, what you have is the long ends property, which means that the ends of this tree are always longer, or at least as long as what lies inside. And if you have these 3 properties, the sequence that you get from this is always a BFB sequence. So rather than taking these counts and trying to reconstruct a BFB sequence, one might think of taking sort of half of these counts and building a BFB tree out of it. And that gives us the first kind of heuristic approach to this problem, which is you start with the top node. There's only 1 node, so there's nothing to do. Then you go to the second level, and now you have 5 nodes. And again, there's nothing to do because they must all connect to the root. And so you just connect them. And I'm sorry, this is not very visible. There's a line out here, so I apologize for the poor translation. Now, because of symmetry, the trees at these nodes will be identical to the trees out here. So we just remove these trees and give these a multiplicity of 2. And then, after that, we could just solve a Diophantine equation to come up with all the subtrees that satisfy the total counts at the next level. So in this case, we'd solve this equation, and that gives you a bunch of different solutions. Again, we are exploring a large space. And then, one particular solution might look like this, for example. And again, I apologize, the trees are not as visible. But again, because of symmetries here, the multiplicity here increases, and all these trees do not have to be considered. So as you go down this tree, because of subtrees being eliminated due to symmetries, you very quickly finish. You have very few possibilities left. And so in practice, this algorithm actually does quite fast. And here's sort of an example that shows how it does relative to the original algorithm. So on a particular count vector segment like this, which had 5 segments in it, this takes only about 12 seconds relative to the simple exponential time algorithm, which took many hours. And so this, for the first time, it gives us a tool with which to look at data and to predict if you can see what fraction lies in BFB. So what we did was something very simple. We just took all count vectors of length 5 and tried to figure out if they supported BFB or not, just to see, is this even a way to distinguish BFB operations from non-BFB? And we find that, if you take count vectors of length 5, only about 15% satisfy BFB. And then we also extended this to count 6, and we find only about 7% can achieve BFB. So if you find a count vector of length 6, one might say, well, this looks like there's evidence that BFB occurred. All right, so is that even true? Well, now if we introduce errors, so again, going back to the admonition from the paper from 16 years back, if you look at the data and you don't consider errors, you can come up with very wrong conclusions. So in this case, if you look at the counts here, they look like this, and this is clearly not a BFB cycle. There's some property which says that all the odd numbers must follow all the even numbers, and this violates that. But if you were to take this and just change the counts a little bit so that the odd numbers follow the even, so the 33 became a 34 and a few other things, then it does actually satisfy BFB. So if you allow for errors, and you make this sort of assumption that as the numbers become larger, the counts become more and more unreliable. So of course you can distinguish between counts 1 and 2 and between 2 and 3, but you can never distinguish between counts, say 25 and 35. It's not possible. And so we came up with a formulation which sort of mimics this, where the count is sort of decided based on, or the variation in count is decided based on the amount of the number itself. We find that a huge fraction of vectors can be explained by BFB. In this case, even if you take the sort of top 1 percentile, 50% of all the vectors, if you take a very small distance, about 0.1, about 50% of the possible vectors can be explained by BFB. So what this says is really that if you allow errors in your counts, you cannot reliably say that something will work off of BFB. There are, however, a few examples, especially those which have a series of alternating 1s and 2s where you have a lot of reliable, where you can say that BFB should not have occurred here. So this is one set of counts where one might say that, oh, BFB is not the best sort of explanation for this. So you can do it in one direction, but not in the other direction. And so we do need a better algorithm. We need something that will allow us to handle larger counts. With counts of segments up to length 6, we find that we are not going to be confidently saying that there's BFB or not. But what if you took larger segments? And now the algorithmic bottleneck does begin to arise. When you have segments as much as 12, then our heuristics don't work, right? And so we had to try this again. And this time we came up with slightly a different direction. So remember, we were going bottom up. We were starting from this and then going in this direction. Now let's start from the top and work our way back. And if you look at the topmost level, all you see are these sequence of blocks, DD, and there's nothing more to do, so we just have these 4 blocks. When we go one level below, we see that there are 6 occurrences of this pair CC, and the 6 must lead to a 4. So what we do is we add 2 sort of epsilon blocks, and now we get 4 blocks which start and begin at C and have a DD in them, and we have 2 of these CC blocks. Right? So that's good. And then we go one more level below, and this time we have 6 but 9 blocks. So when the number of blocks here is larger, we just add extra epsilons and just sort of wrap them, and we get that. And now when we go to the last level, we have 9 blocks at level 2, but only 7 at block 1. So now we need to fold some of these, and now a non-trivial way of folding emerges. So here what we do is we take 3 of these blocks in this particular pattern, and we bring them together and add an A, and we get this special block. And we do this twice, and then we have these other blocks. And so there's a very particular way of folding and wrapping that allows you to do this. And then finally at level 0, we fold all of these into a single block, and we get it. Right? So at a very high level, the algorithm is doing a bunch of wrapping and folding. But as you can see, the folding must be done in a very special way, because you're doing a greedy step. You have no idea what comes in the levels below. If you don't do this right, you're going to fail. And so there's a particular combinatorial idea that I will explain, but I'll leave the details of the algorithm to a later time. So imagine that you have a sequence of blocks, and what we do is we forget about the letters themselves, but just look at the height, right? So you could see the blocks are at different height. This one's at height 1, and now let's imagine that we have a collection in which the highest, they're rank ordered by height. The highest block is say there's 1 of these, and there's 2 at the next height, 4 at the next height, and each collection has 2 to the I blocks at level I. Right? We call this kind of a collection an L-convex collection. And it turns out that when you have a collection like this, you can rearrange it in a very nice way to get almost a BFB sequence. So you take the biggest one, and you put it in the middle, and then you take these 2, and then the next 4, and the next 4. Now, of course, if you read the sequence from here like this, you're going to get a palindromic sequence just because of the symmetry. But of course, this may not be a BFB sequence. In particular, it doesn't have the long ends property. But if you take any such combination and you add 2 blocks at the end, like this, then you get a BFB sequence. And in fact, all BFB sequence at a particular level must have this property, that they are flanked by these high sequences, and in between they have this kind of arrangement of blocks in this L-convex palindromic fashion. All right, so that gives you a new recursive formulation. And then what we do is that in going from layer I to I minus 1, if the number of blocks is larger, then you just add extra epsilon. If the number is smaller, we identify these kinds of sets of convex collections and then fold them. And each time when you fold them, you're reducing the number of blocks by a power of 2. So if you do this and you end up reducing more than what you needed to, just add extra epsilon. All right? But you cannot do them in just any way. And this is sort of the last 2 and the most technical slides. And I'll just roughly go through them. There's a particular way in which you do this decomposition. And what you do is you take all these blocks and you sort of name them. In this case, you have 2 copies of block beta 7, 4 copies of block beta 10, and so on. So you take this as your collection and you decompose this. You'll first look at the odd guys. The odd guys must always be in the center. And so in this case, there's only one odd guy, beta 8. And then you look at the ones that are high, because odd guys must have something high on both sides. And so in this case, beta 7 can surround these, and that'll give you HT. And then you take whatever is left. So this is gone, these 2 are left, you halve them, and then you recurse on each half. So now you have 2 copies of beta 10 and 1 of beta 9, and you kind of do this. Right? What this shows is that if you go along this path, this gives you a convex collection, because this is double the number of copies of this, and that's double the number of copies of that. And so you get a convex collection, and these are the two that flank them. So any collection like this is a kind of rearrangement that you can do. And what you want to do is start from the last end and try to find as many as you can to fold them. And in order to do this sort of more carefully, we have what we call a signature. And the signature, I will not sort of go into how the signature is created, but it creates a lexicographic ordering of all the blocks. And when you do them in the right order, the ones with the lower signature are, if you can fold those, if you can fold something at a higher signature, you can always fold something at a lower signature as well. All right? So if you have two, these are the main theorems here, which is if you have two block collections of the same size, and one has a lower signature, then if you take the one with the higher signature and you can fold it to something of a reduced size n, you can do it for the lower signature as well. So the lower signatures are always better than higher signatures. And the second theorem just says that we can always fold in the way so that we achieve the minimum signature. And because we can do this, a greedy algorithm works, and it gives you the right algorithm. And we have pretty much reached optimality. There's a mistake here. If you have, the input size is k log n, our algorithm is more like k log square n. And if you actually wanted to get the sequence itself, it takes order n steps to write it. So it's kn for reconstructing and k log square n for deciding whether something exists or not. All right. So that's the algorithm in a nutshell. Now the question comes, and you had all these beautiful questions in the beginning, which says, well, how do you apply this to real data? And the real data, as always, is very messy. So one of the things that we do is that we apply this in a windowed fashion, and then for every window, we output, instead of just outputting whether or not it is BFP, we output the distance of the current count vector to the nearest BFP count vector. And furthermore, if the distance meets a threshold, then we accept, and else we reject. And we recognize that these rearrangement operations are happening in the context of many other kinds of rearrangements. And if that happens, then of course they change the counts. But because we allow this kind of a distance function, this is a very general way of getting, if you have low counts, that supports BFP. If you have high counts, you could still have BFP, but maybe mixed with other operations, or maybe you didn't have BFP, but then it will count towards your not being able to identify this. Yes?
What would be typical sizes of A, B, C?
So the question that Pavel asked is what would be typical sizes of the segments? Again, nobody knows. Historically, there are only a handful of examples where they are BFP. I'll show you one example. For those, the sizes are more like between 10s to 50 kbs would be the typical size of a segment. But again, nobody knows. The data, there's a lot of segmentation algorithms, but there are very few where the segments are conclusively shown to have gone through a BFP operation. That's why it's important to formulate and solve some of these problems, to point out of the thousands of cases that we have, are there a few that can support BFP? Yes, Jeng?
So there's no way that BFP on the suffix side will overlap with one on the previous side?
No, we don't. So when you get a data set, you do have, so let me go through some of the examples. We do have diploid, there are two chromosomes, and the counts of the two chromosomes will be mixed even if only one of them is undergoing BFP. So we have a whole bunch of rearrangements that are happening on both chromosomes, but one of them might be undergoing BFP. The BFP typically happens only from the centromere to telomere, at least that's our formulation of the problem. So we take every chromosome and treat it as two examples, one for each of the arms of the chromosome. But again, there can be operations that might do translocations across chromosomes, and those are not BFP. And we also assume that in the BFP that we have imagined, there is no such thing happening. All right, so the BFP operations take place among other rearrangements. And so what we do as negative examples is we simulate diploid chromosomes which have 50 or so rearrangements. Again, the relative numbers between BFP and non-BFP is not known. So we took what we thought are conservative numbers, and we allow inversions, tandem duplications, deletions, and so on. And as positive examples, we take all these operations that are going on here, but in addition, we also do 2 to 10 BFP operations at some point. Okay, and then we create a count vector for the entire chromosome, not just for the BFP part, and that's the data for our problem. And so I just want to show you some results. So for now, ignore the dotted lines for a bit and just look at these solid lines. So if you think of count vectors only of length 6, you can see that even in regular chromosomes, you can identify a huge number of length 6 count vectors that look like BFBs. In fact, at the least cutoff, distance cutoff that you could choose, we have something like a 63% false positive rate. So clearly it's not going to work out. But as you go from count vector length 6 all the way to count vector of length 8 or 12, you can start doing much better. So for example, here we have a false positive rate of 10%, and we get a true positive rate of about 63%. So when you have a long count vector and it has a distance which looks close to something like a BFB, a small distance, then you can be somewhat confident. Now, this number itself you have to take with a grain of salt, because if only, say, 10% of the true operations, 10% of the fraction of true of cases have BFB, then a 10% false positive rate translates into a 50% false positive rate. So if you want to go with lower error rates, say at 1% or so, then you only have about an 18% operation. Okay. So we have to include other data sources too, and here Powell's come into play. So when you have a BFB operation, you also get this minus A kind of junction, which you don't see with other you do see with other rearrangements, but you don't see in excess of these rearrangements. So if you do paired end sequencing and you identify these, then a foldback fraction, the fraction of rearrangements that are of this form, head-head foldback operations, they might be indicative. And we have a composite function that allows you to include these kinds of operations as well. So when you include foldbacks, you can see that the numbers become much, much better. At about 1%, you can already come down to about 60-65% true positive rate in this fairly conservative scenario. All right. So I will say that, you know, with errors and so on, I think it is fair to say that you cannot really do much with short count vectors. But when you have 8 or more BFB cycles and correspondingly large count vectors, and if you use this nice algorithm, then you can really identify cases that are BFBs. And if you use other foldback operations and other signals, that might help in further improving the signal. In fact, if you think about it, there are other much older technologies like fish mapping, which allow you to visualize the spatial arrangement of specific blocks. Even at low resolution, they might add and might improve. So this part, you know, definitely could do with more kinds of different kinds of data, and that's a line that one might think. So here's one example that came out from the Stratton group. This is example of real patients with pancreatic data. They actually sequenced to a high amount of depth 13 individuals, but there's only one where in one of the chromosomes they find a pattern of counts. And when you look at this, you see how unreliable the counts might be. A pattern of counts and a collection of foldback operations, which seem to suggest they claimed was BFB. But remember, they didn't formulate the problem. When you take this data set and you actually apply our algorithm, as opposed to all the thousands of other data sets that we looked at, this one does show up as being significant relative to the simulations that we have done, with the caveat that all of our simulations have artificial numbers, that is like 50 rearrangements versus 8 BFB and so on. So all of that remains open, but we do seem to find cases where there is BFB going on. All right. So I think I want to conclude with two kinds of thoughts. First is, there's this huge body of literature from very prominent scientists which talk about all kinds of exotic structural variations. You know, there's chromotripsis, there's something which is very peculiar, I can't even name this, Fostes MMBIR, and then there's breakage fusion bridge and so on. And they're all proposed based on an observance of data. I think it is incumbent upon us to revisit this evidence and to say, is there a simpler explanation? Can you formulate the problem in a way that you could say that this is the best explanation of the data that they are providing? Because many people, it seems like they just take this and they try to look at data, and rather than trying to see if there is chromotripsis or not, they look at data and say they're trying to see if they find evidence for chromotripsis or not, where where the chromotripsis is not even defined. So that's one direction that I think is plausible, and it's mentioned out here. The other way to think about this is there are many kinds of problems that arise in this notion of genome rearrangements. From the computational side, if you have fragmentary data, you could try and reconstruct the architecture itself. And certainly all the sequence assembly problems, but also more recent directed sequence assembly problems and structural variation analysis allows us to reconstruct the architecture of the sample genome. Then there's the other line of operations pioneered with genome rearrangement, but also in recent work from Shamir and others, which says, well, let's fix the set of operations. Let's say that the only operations that can happen are rearrangements, insertions, deletions, inversions, and so on. Can you take the set of operations and predict either a plausible sequence of events or just the number of events or a distance function like Max pointed out, which takes a sample genome to a reference target? And then there's the third line of questioning, which says, given a sample genome, what are the subset of operations that best explain the rearrangements that happen in the sample genome? And if you can do this, then this problem is solved. But if you cannot do this, then it's worth looking at these problems in their own right. Okay? And I'd just like to conclude by thanking the people who did the work on this. Marcus did much of the experimental work and the initial BFB tree and BFB pivot algorithms. And Shai Zakov is a very smart postdoc working with me, and he worked on the linear time algorithm for BFB detection, which is actually a pretty hard piece of work. And I'd be happy to, it's not published yet, but I'd be happy to share a preprint for those of you. All right, thank you very much. Yes, Ron.
The example that you gave from Campbell's paper drives home the issue, where do you get the counts from? I mean, all your models use integer counts, and the numbers are so, the data is so messy.
Yeah, so the question that Ron points out, and it's a correct question, which is, where do you get the set of counts from? Now, if you look at all the different segmentation algorithms that have been proposed either for array hybridization or for copy number analysis like the CNVR and other copy number analysis tools, they all take genomic fragmentary data and come up with counts. So the right question to ask is not where do you get the counts from, but is the distance. And remember, our algorithm doesn't take a set of counts and say BFB or not. It takes a set of counts and outputs a distance, which is the distance to the closest BFB that you can get from those counts. And the question is, at what point does the uncertainty in count determination destroy the evidence for BFB? And that question can be, that can be answered through simulation. And you can say, if you could measure the counts to this level of accuracy, then you should be able to, and you have sufficiently long count vectors, you would be able to predict BFBs with this kind of performance. That's sort of the line along which we are doing this. And that once we have a sense of what fraction of true cases are BFB and what the count variations are like, we'd be able to say how confident can we be of predicting BFB. And then the other thing I would like to say is that the estimation of count vectors, even though it's still very error prone, is improving by the day. And as you have more and more sophisticated algorithms, I think we will see better count predictions before we see complete sequence assembly. But maybe if you have very long reads, then maybe that won't be an issue. Yes, Paul. Yes, that's actually a very good point. If you imagine sort of evolution of a tumor genome, you could think that every cell has its own sort of rearrangements going on. There I will say, and this is a question that has been raised in the past, usually what happens is that a few kinds of rearrangements, a few members in the cell dominate the population. So when you actually do the sampling, if you have a BFB that deletes a portion or makes a copy, most of the times it's lethal and that cell won't survive. The few that survive, there are a few that are extremely fit, and that's what causes, that's why you have the clonal origin of the tumor and so on. And this happens very quickly. In the 8 or 10 cycles, you have a very rearranged cell. So one could imagine that there might be other individuals, but they just add to the noise of your data, but there will be some that dominate. And well, again, this is all conjecture at this point. We have only a handful of cases of BFB, so as we have more data and we have single cell sequencing data, we would be able to get more clear answers to this question. Yeah.
I was wondering how sensitive your method would be to, let's say, small extra insertions in the middle of this region?
It's not going to be a problem because the way the count vectors are created, insertions are completely ignored in the data. Now that said, sorry?
You take into account
the foldbacks. For foldbacks, it's going to be a challenge. So if you only have count vectors, it's not going to be a problem. Foldbacks, that's why I didn't talk a lot about foldbacks. Foldbacks might seem very attractive, but they're very hard to get, particularly because the junctions are always repetitive. And so you don't have good mapping at those junctions. And if you have insertions and other things, then you will not have. But the way we use foldback is really, we are not using it to reconstruct the architecture. We say, if you classify all the aberrant paired end mapping reads that you see, you can classify them as being evidence for inversion, foldback, duplication, and so on. And we just see what fraction of them qualifies for. So if you miss a few, that fraction will be affected, but it's quite robust from that measure. Yeah. Jane.
Ask a very simple question. I faced this question all the time myself. The formulation of an alternative hypothesis is very important to be able to say.
Yes.
In this particular case, my understanding is you're looking at how many suffix-based tandem converted duplications you make versus arbitrary hyperduplications.
Yes. Yeah. I wouldn't say suffix-based because although it is on the suffix of one chromosome, the other chromosome, the sister chromosome, is also rearranging, and so we actually take all. But that is the way we have formulated this, which is given that this operation is as we have defined it formally. Can you distinguish this happening from in a case where there are other rearrangements going on, versus the alternative hypothesis where it's just the other rearrangements that are happening onto both chromosomes?